So I wanted to give you a quick summary of what I'm about to talk about. So microscopes have evolved significantly over the past decade. So they've grown into essentially a multidimensional tool. They're not just looking at, light. They're looking at, chemistry through fluorescence. You can capture structure, dynamics, and various physical properties of cells, everything from voltage to charge and what have you. There's a whole ecosystem of technologies that have grown around microscopy. And it's the golden age of microscopy, And everything from live cell chambers to all kinds of precision stages, entire palettes of fluorescent proteins, massive supercomputers that are now married to the super microscopes that we have. And what's happening is that they have grown into a powerful tool for aiding the discovery of new therapeutics, right? We're primarily interested in, hey, what is the therapeutic doing to the tissue? Is it inflicting its intended changes? And are there unintended side effects that we want to know about, right? And the biggest problem that we face is that, the images that come out of these microscopes have become more complex, more variable, right? Variability is the bane of this business, right? And massive. We're routinely dealing with single images that are in the multi-terabyte scale, right? So the business of automated image analysis, which was hard enough to begin with, has gotten a lot harder. But on the other hand, this thing called deep neural networks, which is they're all around us, right? They're basically starting to transform this discipline. I just wanted to pick on some examples from our group on the use of deep learning networks to analyze, biological images. I thought I'll pick on two examples, one from a traumatic brain injury. We had a beautiful talk earlier this morning about that. So the response of cells to a brain injury is really confounding because there are so many cell types, they're connected in a dense manner, and it's a computing network. So any damage to this network produces, equally confounding effects. And, like we learned this morning about the microbiome and its effects, there are primary effects from the concussion. There are drugs that have been applied to the patient. The drug has its effects and its side effects and so on, all intertwined together. Now, there are 42 million concussions a year in the US worldwide, and there are still very few good treatments available, right? We're starting to find that combination treatments are more effective than individual ones. So the question is, what can we do to accelerate their discovery? So we started, modifying microscopes, right? So fluorescence microscopes, they can be outfitted. This is a fairly routine Zeiss microscope that can be outfitted with a large palette of filters. So we can, basically capture 10 different proteins at once, right? 10 channels of information at once. Now, we can capture 10 proteins and then, turn them off, then, put down labels for another 10 proteins in round 2, and so on. We can keep going, and we find that we can capture anywhere from 50 to 100 channels or more of data from a single, slice of brain tissue. So some of these molecules basically tell us about structure. Some of them indicate cell types, and others indicate cell status. So we have a very, very rich information about the brain tissue. The next thing we can do is scan across in a brain slice. This is a rat brain slice. And that basically, and we can stitch them together into a mosaic or a montage. And now if we were to do that for all 50 or 100 of these channels, we start to get single images that are in the multi-terabyte range that can, capture anywhere from 250,000 to 1 million cells. And they're rich in terms of information. They can tell us what is the story behind every major cell type in the brain, right? So if you're interested in what a drug is doing, we can not only expect to see the effects of the drug, but also one or more side effects all at once and all in their context, right? So the opportunity is transformational, right? So how do we begin to analyze this? So if you try to analyze even a single image with, say, maybe a 500 or 1,000 cells, we found over the decades that it's hard to do because there is so much variability, right? Just like our faces are so different from each other, cells are very different from each other. They vary a lot, right? What's interesting is that these neural networks have proven to be remarkably capable at capturing this variability, right? I'm just zooming into the result of cell detection. I know you can't see it yet, right? Let me zoom in further, right? Now you see these little dots here? They're the results of cell detections. So if you look at, if you zoom back, you'll sort of see that we've been able to do cell detection on a very large scale, right? With accuracies in the high 90s, right? So now what that means is that now we can look at the other channels and start to infer not only the cell types, but also the cell statuses, right? Just like a human face is composed of eyes, nose, mouth, and so on, these capsule networks are able to resolve parts of a cell in a systematic manner, capture a model of that, and use that to identify cell types, right? And when you run it over large data sets, we're able to tease out subtle pieces of information. Here we had used a combination of drugs, and we found that it had left out an entire subpopulation of cells. So that inspires us to say, can we augment this cocktail with a third component that can help address this missing subpopulation of cells, right? And then what's interesting is that these same neural networks, they come in so many different shapes and forms. We are able to use them to do region segmentations, right? So that we can fit atlases to the brain images and thereby parcellate all these data into little parts. We can also parcellate the data by cortical cell layers. When we do that, right, when we have the effect of an injury or the effect of a drug, we can now tell how it's affecting a certain part of the brain and a certain layer of brain tissue within that region, right? So we are able to narrow down the effects by region and by cortical cell layer, right? So we've been using it in a variety of ways, and we've been using just about every computer vision technique that comes our way. Everything from surveillance techniques like person re-identification. We've been using all these techniques, right? Now, the other example I wanted to pick on relates to cancer immunotherapy. So cell-based immunotherapy, it received a Nobel Prize, and one of the Nobel Prize winners was at MD Anderson here, is our best hope for treating cancer, and yet it only benefits about 20% of the patients, right? When it works, it produces a durable response, right? It produces complete cures in many cases, but it's only applicable for about 20% of the cases. So one of the things we're very interested in is to study these immune cells and their interactions with tumor cells in detail, right? So what we do is that we put them together into a little box. We call that a nanowell. It's about 15 microns wide and 50 microns deep. And what you see in this video is the immune cell, which is on the lower right, okay? It goes and actually forms a bond with the cell in the middle, and the red color that you see here is a cytotoxin. It basically bites that cell, delivers a cytotoxin, and kills it, right? It does it with incredible specificity. Now, we wanted to scale that up, so we did. So basically, we can now start to see these phenomena, right, play out over literally tens to hundreds of thousands of nanowells, right? So you can form a chip made of PDMS by my colleague Naveen Varadarajan. And basically, what this is able to do is that it's able to reveal, these dances, right? So there are basically cellular dances that are taking place in each nanowell. I don't know if some of you remember this movie, this Alfred Hitchcock movie, right? So there's this gentleman sitting in a wheelchair who's looking at an apartment block in New York City. And in every window, there's a new story taking place, right? So that's what we're talking about. But we're looking at basically tens to hundreds of thousands of these stories taking place, right? We want to know in which of these nanowells killing happened, right? And what were the details of that killing, right? So now what's interesting is that all these years we used fluorescent markers, because fluorescence is their superpower, right? We use these fluorescent markers to delineate the cells, to highlight them, to identify cell types, and to identify the cell status. So when a cell is dying, right, this blue marker basically lights up, right? And not the blue one, but the yellow marker basically lights up. So deep neural networks, again, have proven to be our ally in terms of being able to, first of all, identify all the different nanowells. Some of them are diagonal or rhomboidal nanowells. We are also then able to track the cells over time using long short-term memory neural networks, right? We're able to essentially tease out measurements of when the biting happened, when the neurotoxin was delivered, and when the cell actually died, right? So now what's interesting now is that with the advent of these neural networks, our work has entered a new phase, right? What we are starting to find is that increasingly, we no longer seem to need these fluorescent markers. So there is one channel that's available, which is a phase contrast channel. This is a black and white channel in the microscope. And this channel has been hard to analyze with computer vision algorithms. But now, these neural networks, fairly routine ones, like mask-RCNN, for example, in this case, we're able to actually identify not only delineate the cells, but also identify what cell type it is. And we've started to develop a very long list of capabilities, all without the benefit of fluorescent markers. We're able to identify cell types. We're able to identify nanowells of interest where certain types of actions have happened, right? And basically, we're able to, of course, map out cell movements, cell-cell interactions, like the biting of cells. We're able to identify critical events like cell death, formation of immune synapses, okay, formation of apoptotic bodies, detection of cell division. All of these things can now be done, okay, from just the phase images. What's interesting also is that we're finding that these phase image analysis, we kind of started out thinking that it's going to be inferior to the fluorescence-based image analysis on which it was trained. But the opposite turned out to be true, right? So the phase image analysis has become adept at identifying cell death well before the fluorescent marker kicks in, right? So for example, the fluorescent marker kicks in, at this 15 minutes, whereas this cell was identified as beginning to die much before that by analyzing the phase channel. Why does that work? It's kind of intriguing, right? And so I thought I'll play a few movies to show you what's going on, So let me see if I can play these two movies. So the video on the right is the cell death marker. It's a fluorescent marker that suddenly comes on alive once the cell is determined to be dying or apoptotic, right? But if you observe the video to the left of that, right, on this side, you can sort of see that there's a lot of structural or morphological cues that the cell is putting out, right? And that's what this algorithm is cueing into, right? So let me talk a little bit more about that. So when a cell undergoes apoptosis, right, it actually undergoes a lot of shape changes called blebbing, and then it puts off little parts, almost like a bomb going off. These are known as apoptotic bodies, right? So we're now able to train neural networks that can screen all the nanowells. You've got the 10,000 nanowells. It can screen out which nanowells have apoptotic bodies in them, right? That's a fantastic capability to have, right? So we can now narrow our attention down to just those nanowells. The next thing is we're able to actually delineate these apoptotic bodies, or do segmentation, right? And then we can spatially associate them with the nearest cells, and that tells us which cells are apoptotic. So what's beautiful about this is that this capability actually works better than the fluorescence-based method, because in the lower video, I don't know if you noticed it, the fluorescence-based marker just didn't work. And yet, the label-free method actually is able to detect apoptosis successfully. This is an event of huge importance to this type of research, right? So now what's also interesting is that these networks are capable of cueing into sub-visual cues. For example, we can tell where the nuclei are in a cell, even though when I look at the image, we can't see them, right? So basically, we can computationally infer the locations of cell nuclei from a label-free image. Now, there's a body of literature now that indicates that we can potentially infer many more structures, mitochondria, endoplasmic reticuli, and so on, right? And if you're doing high-speed, high-throughput microscopy, right, you can lose focus very easily, right, to the slightest of factors. Now, increasingly, something called like deconvolution. This is something we used to do in the 90s with a lot of physics-based modeling of the point spread function and sophisticated EM algorithms for deconvolving them. These neural networks are able to be trained to do that. And when we use them, we're finding that the cell analysis goes much better, right? So in the case on the left, right, the cell segmentations are much more reliable. When the cell segmentations are more reliable, we can determine cell contacts, which is an event of huge importance to us, much more reliably with fewer false detections, right? Anyway, to summarize, I've shown you two examples where these neural networks are having a transformational impact, right? I used to despise them at one point. Now I've embraced them, right? So in the first case, right, we see there seems to be a trifecta at play, right? Multiplex fluorescence, wide area scanning, and CNN-based tissue profiling is basically in the process of transforming histology. In the second example, right, we can process massive video arrays, right? And increasingly, we're able to do it in a label-free manner. That opens the door, because in these instruments, there are very few fluorescent channels available, 3 or 4 maybe, right? We can use these precious fluorescent channels to do new discovery of new markers, right? Anyway, it's an exciting area of progress and promise, and I'm happy to answer any questions.
Thank you. You hinted about progress in subcellular structures, position of the mitochondria. Are you optimistic? You think it's a smooth trajectory?
There was a paper, not from our group, but there was a paper in Nature that came out about 6 or 7 months ago describing this technique. So it gives great room for optimism here. The question is, can we do that at high throughput? All of their work was done in low throughput, where you can do exquisite, high-resolution imaging. But here at high throughput, we're happy to just get a non-blurry image, right? So there is going to be some limitations, okay, at high speed, high throughput, but I think we can do more, go beyond nuclei.
One last question, yeah.
So you had a cross-section of fluorescent cell image in your first example. Did you obtain that from a living person?
These are from sacrificed lab rats. Yeah. These are rat brains.
So you're not able to use that as a diagnosis?
No, this is for drug discovery. So that's the end game here. So.
Let's all thank Badri again.